it is my honor and my pleasure to bring the most requested doctor in Highwire history. I'm talking about Dr. Andrew Kaufman is joining me now. All right, Dr. Kaufman, I mean everything I said. We are literally getting lambasted on YouTube. When are you going to finally talk to Dr. Andrew Kaufman? Are you afraid of Dr. Kaufman? Because we are just, as I said, firing and shooting from the hip here, what is your background that makes your voice important enough that so many people are requesting you? So just give me a little history of where you come from and what your education and specialty is.
Sure. Well, I studied biology at MIT as an undergraduate, where I first learned about research. I worked at a couple of different biotech companies. I was a physician assistant, and I worked in cancer medicine, mostly in hematology. And then I went to medical school and did my psychiatry residency at Duke, and then subspecialized in forensic psychiatry in Upstate Medical University in Syracuse, New York. And I joined the faculty there and was on the faculty for several years. And I conducted research that was published in peer-reviewed journals. I held leadership positions at the university and in my professional organizations. And I even had a biotech company where I invented a medical device and tried to make that a commercial success. I've also testified in a lot of legal matters as an expert witness in various different courts. So I've done quite a lot of things in healthcare and medicine, and had a wide variety of experience. I think what is unique about my perspective or my position here is that I don't have any ties or obligations to either the medical or the scientific establishment. But I do have the skills to understand scientific research and read papers. So I can report on this and express my opinion about what I believe to be true without having any repercussions or pressure.
Excellent. All right. So, you know, for those that have been watching the Highwire all the way through this pandemic, we have laid out real concerns, beginning with the modeling around this pandemic and the supposed deaths that were supposed to take place. Very early on, it became clear to us that this virus did not have the death rate we were being told. I would say that in many ways, we were one of the first media organizations to predict the fall of the imperial model, which, of course, it came crashing down. We have been calling out, you know, Anthony Fauci and Deborah Birx, who have continuously kept changing their story, readjusting numbers based on models. We know that we have bloated death numbers because of, you know, the way that we are reporting deaths in hospitals. All of these things we've covered. I've talked to scientists that have been on here, like Dr. James Lyons-Weiler, who at first said that this was a man-made virus, potentially a bioweapon that could have escaped, then decided, he came back and said maybe it's natural. Zach Bush, who said he thinks that maybe this is more likely to be natural. But so we've covered the gamut here. But I would say that we have been questioning the mainstream narrative from the beginning because it just doesn't add up. But you are actually taking this, I think, a step further than the Highwire has ever really gone before on this topic, because up until now, we are using the idea that this is a virus that has swept the nation. Maybe it was here earlier than we thought. The numbers aren't what we seem, but there does seem to be some dangers when it gets into nursing homes, all of those things. You are actually questioning whether or not COVID-19 exists at all. Am I correct about that, or please clarify your position?
Yeah, thank you. That's pretty close. So I just want to separate the SARS-CoV-2 virus, which is what they've named, what they call as a virus, and COVID-19 being the illness. Okay. So I actually reject both hypotheses, but I don't want to come across as saying that I deny that there were people dying during this mortality spike from late March until May. I just have different explanation for what resulted in those deaths. But what I've done from the beginning, once I noticed that there was some fishy things going on in the policies of how they were handling this pandemic, because it was divergent from any past policies that I'd ever been familiar with, I went and looked at the papers that had claimed to isolate this virus, and there are essentially 4 papers. And when I read these, I read them very closely, and I had to, do some investigation because they use kind of a lot of language that's difficult to understand unless you're publishing in this field. So it took me a bit to parse out exactly what they did, but what I found out is that they never isolated or purified a virus. So what I mean is, in order to identify a virus, it's basically a particle, right, that has a membrane and inside of it some genetic material and a few proteins throughout. And so in order to identify that, what you would do is that you would basically purify it from a sick person, from the diseased part of their body. In this case, they took samples from lung fluid because the people that they identified as having this illness had respiratory symptoms. But they never actually purified any viral particles out of that lung fluid or out of their tissue culture experiments later on. Instead, what they did is they amplified a piece of genetic material that had a part of its sequence that they were specifically looking for because they had pre-identified these sequences as being from viruses. And they just basically identified a snippet of this genetic material and said that this was a virus. And they did really nothing more. So there was never a particle that was purified from which they would extract genetic material and say, this RNA came from this particle, therefore it belongs to the particle and it makes up a basically genome of a virus. If they wanted to go a step further and prove that such a virus would cause an illness, what they would have to do is then put that virus particle in a healthy host and then show that that host develops the same disease, and those are called Koch's postulates. And that's basically the common sense rules which the germ theory scientists originally formulated themselves to prove if a germ or pathogen causes an illness. And this was never done either. And this has far-reaching implications because the test, the main test that's been used, the RT-PCR test that I'm sure you've talked about at length, is based on this little piece of RNA that we don't know where it comes from, because it came from this lung fluid, which has many, many things in it, many sources of RNA, DNA, many different kinds of cells, including human and microbial. And we also know that many what they call viral sequences have been found in our own cells and particles that our own cells make, known as exosomes, in the past. So it could very well be that this RNA is from our own cells, from exosomes, or it could be from another microbial source. We just have no idea of telling where it was because they never found the particle that it came from and took it from that particle. And there's a new article that came out written by Torsten Engelbrecht where he actually emailed the authors of these 4 papers and asked them directly, did you show any evidence of a purified virus? And all 4 of them answered that they did not purify any viral particles. So they're essentially admitting that they don't know the origin of the genetic material.
So just to be clear, this is something they could do, right? It is possible to isolate this particle and figure out where it came from, what it is. Why would they skip what seems like a really important issue? And look, if it was just a common cold and they just wanted to get to some other hypotheses and we're doing some studies in a lab somewhere that I don't care about, fine. But you have shut down the world. You have destroyed the economy of the United States of America and many other economies, most of the economies around the world. You would hope that the best science was being done before you took that step. So how hard is that step? I mean, is it like cost $1 billion to isolate it, or is there a reason why they would skip what seems like an obvious step to make sure we knew what we were talking about?
Yeah, well, that's a great question, Del, and this is why it's so important, is because all of these measures that have been taken are based on this scientific assumption that has not been proven. So they would actually be able to do this as long as there were an actual viral particle that they could find. So the procedure to purify these particles has been developed long ago, and it's been used many times. So it was originally developed to isolate these viruses that live among bacteria cells called bacteriophages. And a lot of the science is based on the science of bacteriophages. But they've also been able to use the same technique to purify exosomes, which are particles very, very similar to what they say virus particles are, that are made by our own cells in response to some kind of illness or toxin or infection. And so the procedure is essentially that they take the body fluid that contains the particle, and then they would filter it for size to remove any bigger cells, because these particles are much, much smaller than any human or bacterial cell. And then they would use several different techniques using the centrifuge, which can separate things based on density. And they would essentially, through this process, would purify the viral particles into a tight band in a test tube, and then they can pull them out with a syringe or a pipette. They can look at them under the microscope and see a pure homogeneous particles. They could then also take out the genetic material and have a full sequence of it from end to end, from one piece rather than from thousands of pieces. And they can also characterize the chemicals on it, like we've been told that there's an angiotensin II receptor, right? So they could do an assay and show that it's on the membrane of this particle quite easily. So this science has been laid out. It's been done for many other types of experiments, like I said, with viruses and lower organisms as well as with exosomes. So there's no reason it couldn't be done. It might be slightly more expensive. It takes slightly longer to achieve, but they could, you know, first find these genetic snippets that they theoretically think may be associated with a virus, but can't prove where they're from. And then they could publish that as a preliminary report and then follow up with the actual purification where they can prove, you know, beyond a shadow of a doubt that this is a virus.
So
and if they did that,
to be clear,
yeah.
Since we've been looking at this since, you know, last December, we've certainly had enough time to have done this science by now, right? We should be able to clarify these things.
And if you are taught how to observe, you cease to observe.